Yeah, okay. I've stopped sharing.
Okay.
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 Hand it
Hi.
over to you.
All right. Thank you. Hi, everybody. Welcome back to Singapore Healthcare Engineering Forum 2020. So we should start with team two of our event right now. So the team two, second team of the event is on diagnostics. We have two very exciting speakers lined up for you. Let's start. So the first one is Dr. Anthony Chua. Dr. Anthony Chua is the CEO and co-founder of the Singapore-based startup called StratifiCare. So StratifiCare is involved in the developing of a dengue test prognostic for dengue tests. So he's going to talk about a very interesting topic, which I'm sure what many academic scientists might have in mind. So his topic is on their experiences in crossing over to industry as academic scientists in IVD development. So over to you, Anthony.
Thank you, Gordon. Okay, can everybody see my screen?
Yes, we can.
Okay, great. Right. Hi, good morning, everyone. I'm Anthony, the co-founder and CEO of StratifiCare. StratifiCare is a Singapore-based startup founded with the vision of empowering personalized medicine for tomorrow through our innovative diagnostic solutions. And so before I start, I would like to first thank the organizers, BES, IEEMB, IEWIE, and also IES BETC for inviting me to share my experience on our of us crossing over to the industry as academic scientists in vitro diagnostic development. My sharing session today will briefly follow these outlines. First, I will share a bit on our experience on working on translational medical technologies at the National University of Singapore. This will be followed by sharing on experience on the commercialization of the severe dengue prediction technology. And lastly, I will also share further on how inventors can develop medtech innovations further in line with industry requirements so as to make the technology more attractive to potential industrial licensees. For me, it all started back in 2006 when I went over to the Philadelphia University of Pennsylvania under the NUS Overseas College program. And during that time, I was trained in high-tech entrepreneurship by lecturers at the Wharton Business School. And at the same time, I was working at a pharmaceutical startup there in Philadelphia, developing antibiotic candidates as a last line of defense for multi-drug resistant bacteria. Later on, Protest Pharmaceutical was acquired by Novartis. After the one-year NOC program, I returned back to NUS to continue and finish up my BSc, my Bachelor in Science. I've joined the Department of Microbiology, specifically under Professor Mary Ng, to do my honors, followed by my PhD studies. During that time, for our research group, we were pretty well known for our ability to express and produce purified dengue and flavivirus structural protein. And this led to quite a few collaborations. Because of my prior experience and interest in doing technology commercialization, Prof. Mary have involved me in quite a number of these projects. The very first is with S3 Austria, whereby we generated hybridomas that produces neutralizing antibody against West Nile virus, which is one of the cousins of dengue virus. It's really a proof of concept project whereby we try to encapsulate these live hybridoma cells in capsules and grow them in vitro to see how they fare and whether or not they can continuously elute therapeutic antibodies. The ultimate goal is to implant them into humans, into patients, whereby these implanted capsules can continuously produce and release therapeutic antibodies, and the patient don't have to go back to regular jabs of biologics. At the same time, we were also working with MP Biomedicals. We provided them with purified dengue proteins and for them to coat them onto lateral flow test kits to develop their like a rapid test diagnostic test kit for dengue. At the same time, we also are part of the team who attracted Fujitsu into Singapore to set up their first biomedical laboratory, which later spin off as Aptar Biosciences. For them, they are mainly working on aptamer technology, whereby they, for both diagnostic and therapeutic purposes, and together with us, they are looking at how aptamers can be used as a dengue diagnostic tool and also for potential therapeutic. So one thing in common between MP Biomedicals and Fujitsu is that we really produce the protein and then threw it over the wall for them to continue with their development work. Back then, we were pretty like just academic scientists. We didn't think much, and then we also didn't think about really going to their place to learn more about the development process, which like retrospectively, I thought that it would be a pretty good idea. Then in 2008, our lab became involved in the NMRC Stop Dengue grant, whereby we are working with our main local public hospitals and all the research institutes and universities to come up with solutions for the dengue problem in Singapore. One of the main problem statement that the clinician brought out is the over-hospitalization of dengue patients due to our aging population. We need more and more hospital beds reserved for our elderly patients, and they see that quite a huge portion, about more than 50% of the patients hospitalized actually do not go on to develop severe complications. And thus, we are tasked with the job to identify biomarkers that can accurately predict which of these dengue patients will go on to develop severe complications and really require the hospitalization. We successfully identified the biomarkers, and then the first thing is to file a patent. Back then, again, we were like, don't know much, and under which countries do we exactly have to file in? So we asked around, don't have a good idea, and then with the help of ILO, we filed in some of the jurisdiction. Then after the Stop Dengue grants ended, we went on to apply for the NRF Proof of Concept grant to further develop the prognostic test or the severe dengue prediction test. By the end of the grant, we were very happy because, yay, we have a working lab prototype available already. So during this time, we were also reaching out to all the potential licensees, like the big companies in the working on IVDs, to see whether they are interested to collaborate with us or to license the technology. But the common feedback that we've gotten from all of them is that, oh, you guys are too early. But we were like thinking, hey, but we've come out already with the lab prototype, a fully functional lab prototype. I've also tested it with clinical samples. What do you mean that it's still too early? And so we were very like puzzled, and we don't know better. And so when nobody wants to work with us and we still see the value of this test, we decided to move forward on our own. That's where we incorporated StratifiCare, and we applied for the SG Tech grant with NUS, with Enterprise Singapore. So, but after this whole journey, looking back now, if you were to ask me, I will also say that we are too early. And today, the main objective of this sharing session is to share with you why this is so, yeah. So we are developing the world's first severe dengue prediction test, StratifiCare. It is a state-of-the-art biomarker immunoassay with unparalleled accuracy. We are offering accuracy of 90% sensitivity and 97% specificity. For each of these ELISA tests, we can run up to 8 patient samples at one go, and we are able to do the prediction on day 1 when the patient comes to the hospital, which is usually day 3 to 5 after the onset of fever. Currently, what's happening is that when a suspected dengue patient goes to the hospital, his blood will be drawn and he will be tested for dengue. If the blood test comes back as positive, the doctor will screen the patient's symptoms against a list of warning signs as sort of like listed by WHO. These warning signs include pain around the eye, abdominal pain, continuous vomiting, and so on. And so long the patient exhibits one or more of these warning signs, he or she will be deemed to be at risk of developing severe dengue and immediately hospitalized. But retrospective data will show that more than 50% of all these patients are unnecessarily hospitalized, and this increased to unnecessary healthcare expenses. This is important because for the patients who don't have healthcare insurance, they are paying out of pocket and paying for nothing. For the insurance companies, it's unnecessary reimbursement for their healthcare policy, and for the government, which is substantially subsidizing healthcare for us, is unnecessary use of taxpayers' money. So with StratifiCare, our ideal, what we aim to achieve is really after the blood test comes back as positive, the doctor will order StratifiCare again to test whether or not the patient will be at risk of developing severe dengue. If so, of course, the patient will be immediately hospitalized, and with a better triaging method, the doctors and nurses will now be less overwhelmed, especially during a huge dengue outbreak. They can have more time to focus on the patients that's truly predicted to be at risk of developing severe dengue and provide timely, aggressive, supportive treatment. On the other hand, patients who are tested to be not at risk of developing severe dengue, they can be managed as an outpatient, and they can recover in the comfort of their own home by themselves, and this will help to reduce unnecessary healthcare expenses. All in all, we are looking at up to 7.2 hospitalization days being saved and a global social economic cost of $7.6 billion. And because dengue mainly affects developing countries, the bulk of these savings will go to developing countries. This is our team. Together with me, I have Keith, who is our co-founder and COO. After he graduated from NUS, he went on to work for Merck, and he currently heads the commercial marketing team at Merck for APAC. So he's the expert in product commercialization and marketing. Tiffany, she joined us earlier as a collaborator from Nanyang Polytechnic, and after we spin off, she joined us as a co-founder and our chief regulatory officer. She was certified by the British Standards Institute as a certified lead auditor for ISO 13485, and she currently audits for one of the top 15 medical device companies in the US. Of course, last but not least, we have Professor Mary Ng, who is both Keith and my PhD advisor. She's also the co-inventor of the technology. She has been working on the dengue virus for more than 30 years at NUS, and she previously sits on the Dengue Advisory Committee for WHO and was an early advisor to the Novartis Institute of Tropical Disease. Along the way, we've also recruited very experienced advisors. Lisa and Ted, they are both serial deep tech and medtech entrepreneurs with 1 IPO and 2 exits under their belts each. Ted in particular, he has been working with US FDA for the past 20 years on the registration of various devices. In view of the social impact that we can bring to dengue patients, especially in developing countries, we have recruited Professor Radekop from Erasmus University. He's an expert in health technology assessment and has helped numerous companies and startups in Europe run their cost-effectiveness studies. So what really sets StratifiCare aside from a lot of other medtech companies, medtech startups out there is really that we decided to set up our quality management system right up front from day one. Why we decided to do so is that at the point where we were about to spin off, we started to speak to some of the regulatory experts. We know that regulatory will definitely come in as we are a medtech company, but it's still a fact-finding process. And one of them really, his comment struck us that he told us that how do you even know how to properly develop a medical device or IVD test if you don't have your quality system up? We were like, what do you mean? We've already set up our working lab prototype already, right? What do you mean that we don't know how to develop our test properly? But as researchers, we thought that it's always good to do things right up front than to leave it to the end and then have to repeat 2 years worth of work. So we thought that, okay, let's do this upfront and set up our quality system. And lo and behold, we were so wrong then. Yeah, it's totally the right thing to really start working on your quality system right from day one. And so we did that with the intention of setting up a robust quality system to develop and manufacture medical device that consistently meet customer and applicable regulatory requirements for safety and performance. These are the two main things, safety and performance, that regulatory authorities always look for. And together, we spent more than $50,000 just to set up the system and to train ourselves on the system. Of course, we are certified for ISO 13485, which is the quality system for the development and manufacturing of medical devices. And for our case, we've also set up our quality system in compliance with 21 CFR Part 820, which is the requirements of the US FDA, and also 9879 EC, which the in vitro diagnostic medical device directive, which is in compliance with the requirements of the European Union. For IVDD, here is going to be obsolete soon, so currently we are doing the gap analysis to bridge over to IVDR. And it doesn't just stop there, because with 13485, it brings along a lot of other ISO standards. One of them is 14971, which is risk management for medical devices, and also other small, more specific ones. So there's really quite a lot of framework to follow. And we also decided to hire the most stringent certification body, at least in Singapore, to audit us. There's a whole many choices for you to choose from. And why we go for the most stringent one is because we wanted to really learn and establish the best practices from day one. So if they were to come out with some findings, we will be able to quickly make the adjustment and implement them so that we are doing really the best practices in the industry. And so we are very proud to announce that for our initial certification back in 2017, we have only one finding. All this credit mainly goes towards Tiffany, who's our Chief Regulatory Officer, because she's the main person overseeing the whole quality system. And with her very like on the ball and her vigilance, we didn't have any findings for our two subsequent surveillance audit. And for the recertification audit that we just had this year, we also have no finding, which is a huge thing, a huge achievement for us. And so now when we speak to the big boys again, nobody have like came back and tell us that, oh, you guys are too early again, because we literally speak the same lingo as them. What's verification, what's validation, there's specific definition to them, what you're supposed to do, and they know exactly where we are and we know exactly what they want. And so what exactly is a quality system? It's really a framework whereby you identify the inputs, put it through a series of processes and churn out the output. Inputs can come from business, can come from customers or end users, or even from regulatory authorities. And for the processes, there's a lot over here. And eventually we wanted to really churn out outputs in the form of a diagnostic test that can meet the accuracy, the safety, the requirements of the end users, the patients, and also the requirements of regulatory authorities. And so the main gist of the whole quality system is really in these processes. Things like risk management, you have to look into calibration. We all came from the academic background, so we know that calibration of pipettes probably is the last thing that you will do unless you have a lot of like excess funding, grant funding that you have nowhere to spend, that you send for calibration. PM of equipment is the same thing, and all these form part and parcel of like the quality system, and is a requirement of regulatory process. So in a way, if we don't get this up to mark, it's very hard for any development work in academic setting to be recognized and readily to be recognized by the regulatory authorities, which means that the industry licensee, they will also have to repeat all this all over again, which makes them less incentivized to license the product earlier on. And for equipment wise, we also have to do things like installation and operational qualification, which like a lot of people don't really know how to do. And after we've done IQOQ on these instruments, did a PM and calibration, we really want to make sure that only our own people who are trained to use this can work on it and can use these instruments, and other lab other lab members who are not trained, they will not have access to them. This is a challenge, more and more of a challenge, especially at NUS or in academic institutes where the lab has become more open concept to encourage cross-fertilization of ideas. So design control is really the method to control the design development process of new or even modified medical devices, and so as to ensure that they meet the needs of users, patients. Yeah. And so what is considered medical devices? In view of the time constraint, I'll probably skip through this. If anybody is interested to find out more, I can definitely share later. One of the things that I would like to point out is that a medical device software, a software can also be considered a medical device. Things like a software to really read radiogram or MRI pictures, they are definitely considered medical software. And in addition to ISO 13485, they also have to comply to IEC 62304, which is another standard. So for StratifiCare, we mainly split our design control processes into 5 different parts: initial phase, prototype phase, pilot production phase, design transfer phase, and pilot launch. And in the these phases vary slightly from organization to organization. And for us at StratifiCare, from our experience, it's really the initial phase that can be readily be conducted in academic setting. Once you move into prototyping phase, it's more and more difficult. So what is involved in the initial phase? For the initial phase, is a phase where the product is initially defined on market needs assessment and how, or if it is a new product, a modified product, how it's supposed to be improved. During this phase, we have to set up the design history file, which will keep record of all the development processes and the results, the data, everything right from day one, the product is being conceptualized, all the way until the product becomes obsolete and is pulled off entirely by the product owner from the market. So in a way, it's the whole life cycle of the medical device. Then we are, you also have to do design development planning, and more importantly, identify what are the design inputs from the end users, such as patients, healthcare professionals, and build them into your, record them down and build them into, design them into your medical device. Then you also have to think about what are the risks involved that can come out, that can arise from your product, like to the patients in terms of the design, or to the users during the process of it being used, or even during the manufacturing process. And of course, the regulatory strategy, which countries are you going to sell your product in? You have to identify it upfront and then look into the requirements, because the requirements differ from one regulatory authority to another. And in a way, you have to build all these requirements into your design inputs right up front. Thereafter, you have to do some feasibility testing to test whether or not your design concepts actually work. And all this testing will have to be properly controlled, recorded in controlled lab notebooks or formal test protocols. In a way, we can't really think that, oh, today I come to the lab, I think that I'm going to modify this particular variable, I just go and do it, and then I record down my result. No, we can't do that. You have to first write down the aim, write down the materials and methods, and then what's the expected outcome, and then you get somebody more senior than you to really verify and sign it off first before you conduct the test, and then later on record the results, interpret it, and get it signed off again. So everything is very formalized, and all this, the regulatory authority will want to see them. And so next will be the pilot production phase. This part is pretty difficult to actually do in-house in the academic setting because of the instruments that's required to be used. And as mentioned earlier, it's very hard to do IQOQ or do proper PM and calibration for common shared equipments. This is also the phase whereby we start to generate design outputs, and we have to identify the risks involved in the design and try to come up with methods or change the designs so as to reduce the risk as much as possible. and also evaluate like what kind of risk can possibly arise when the user is using the medical device. And the one big chunk of activities that you need to do during this phase is really design verification, where you have to go through all the different tests to make sure that your product actually meets your predefined engineering specifications. And all these tests again need to be statistically analyzed, and the sample size of the test have to be predetermined. Some other things for that we usually don't consider in the academic setting will be transport simulation studies, shelf life, in-use studies, and also for the implantable medical devices, sterilization, and biocompatibility. And last but not least, you have to think about how you're going to manufacture it. So looking into manufacturing flow, quality control points, what are the manufacturing input requirements. For the rest of the 3 phases, it's really next to impossible to run it under academic setting. So I will leave it for now. And so, yeah, I've pretty much come to the end of my sharing session today. And like because we've crossed over from academic scientists to the industry, we are really open to help other fellow academic scientists who are really either trying to push their new innovative technology out for licensing or thinking of starting their own company to come and talk to us, understand a bit more about the requirements of the quality management system and how to really develop medical devices properly according to industry standards. These are my contact details, so feel free to reach out to me anytime. Thank you.
Hi, thank you, Dr. Anthony Chua, for your interesting talk. Yeah, I'm sure you guys have much to learn in the beginning when you started out, trying to commercialize this thing, and the journey has been quite interesting. So we will proceed with Q&A for Dr. Anthony Chua after, during the Q&A session. Right now, we have our second speaker, Dr. Weng Ruifen, online. So Dr. Weng is the Vice President of DXD, Diagnostics Development Hub, which is part of A-Star, where she drives the product development and project management there. And her topic is on the diagnostic development under COVID-19 pandemic. Ruifen, are you online?
Hi, Gordon.
Oh, hi.
Thank you very much for the introduction. I'm trying to set up my slide. Are you able to see the screen?
Yes, we can.
Let me see whether I can turn on my. Hi. So thanks a lot for the introduction. And hi everyone, I'm Ruifen. So as Gordon has introduced, I actually oversee the product development and the project management teams at the Diagnostic Development Hub, or DXD Hub. So DXD Hub is a national platform for the productization of specifically diagnostics solutions. It is funded by the National Research Foundation of Singapore and hosted by A-Star. So thanks a lot for the invitation. I think it's a pleasure to be able to come here to share some of our recent experience developing on various diagnostic solutions for COVID-19. So I think maybe I will start by going through the process of product development of a diagnostic test. So if you look at the process, this variation of what it takes from the lab to the market, we always actually start from the discovery, of course, and then it goes through this process called translation, and then the whole process that we often refer to as productization. I think most of us are actually familiar with the term product development. I think most people refer the whole process of productization as product development. And I think at DXD, we would like to call it productization, just to differentiate that a little bit from translational research. And then of course, at the end, market adoption. So what DXT has built and has been focusing on in the past few years is really on this segment under productization and market adoption. And that's actually linking the output of R&D from translational research to all the way to the market. So at DXT, we work with R&D output coming from both private companies as well as the public sectors. We take this R&D output downstream towards productization, and then we connect the output of this process, typically in the form of a product or a service, to the end users. So for diagnostic products, most of the time, the end users will be operators in the clinical diagnostic lab, or actually the clinicians directly. So what goes on in between is this V shape that you see at the bottom of the productization and market adoption. In this process, what we start with is always user requirement and user requirement. User requirement is an extremely important element because it defines how the product should look like. And when we actually take it out to the market, how is the product going to be used? And somehow the user requirement define more or less the use case for the product that you are trying to develop. So we are really not talking about a snapshot of the test. In fact, we often call it the anatomy of a diagnostic test because it always involves the whole diagnostic workflow. I think this is something that I will elaborate further in my subsequent slide. So we focus on these two elements in the red box here, the end-to-end productization process and the agile iterative process. So by end-to-end productization, basically we means that we start with the end in mind. We actually go out and look at the usability of the product or during conceptualization. We talk to the end users, we see what the product should look like, and then we go back to the beginning of this whole process. So you can see in those boxes at the beginning of productization, it actually says functional prototypes. So by functional prototype, it's not just about the prototype itself, but it's also about how the solution actually works with as a functional unit. And then system integration. I think it's very important to know how, for example, how the assay will work in whichever system that you envision the solution to be deployed. And we need to be able to actually integrate the assay onto a certain platform most of the time and a certain system. And that's what we call system integration. So we start with that, and then we actually go down and perform the analytical verification of the whole system very systematically. And all that will be done before we actually go down to clinical validation. So at DXT Hub, we always like to advise our partners, our collaborators, that we actually need to be able to understand as much analytical functions of the product as possible before we actually go down to the final stage of clinical validation. So I mentioned the anatomy of a diagnostic test. What do I really mean by the anatomy of a test? So if you look at this, when you look at diagnostic tests, it's never about just the assay, which is just really one of the many components of the whole diagnostic solutions that you are trying to offer to the end users. A diagnostic test actually starts from when specimen is collected, because you always need to consider what specimen are you testing. Are you testing blood, saliva, or nasopharyngeal specimen, particularly in the COVID situation? And then when you think about a diagnostic assay, we always will start with what is the sample type or specimen that we will use, what is the sample collection procedures, and how samples should be stored and transported. Also about the stability of the samples and so on. So I think all of these are actually very important pre-analytical consideration during assay optimization. And then we think about the assay itself. And as we are looking at the assay, that would considerations are such as are we looking at an assay that will detect nucleic acid, or are we looking at the assays that will be detecting the proteins, or are we trying to identify some sort of profiles that has a good correlation with the disease that you are trying to detect. So after consider what types of assay that we are going to look at, depending on what analyte you are going to detect, we then need to think about what system the assay is going to be running on. For example, if it's a PCR assay, there are already so many different types of PCR platforms on the market. And there is really no reason to try to reinvent the wheel or to build another PCR system. Unless, of course, if your PCR system offer unique features that the available system in the market is not able to offer. So then you actually will want to consider what type of commercial PCR system you will want your assay to be working on. And actually it can be all of this. But then every time when you use one specific system, is a whole new process of actually analytically verifying the performance of your assay on that particular system. So that's why when we always thought about everything as a whole rather than just the individual assay that you are trying to register with HSA or with the US FDA. And we actually need to think about if this is going to be a lab-based test, what is the system that the lab is using? And are we looking at the high throughput type of test, in which case every time the lab is going to be running 96 samples in a row? Or if the lab is a low to mid throughput type of lab, and then where each time they will probably just run a couple, 5 or 10. So it's important to consider that, because all of this has a huge impact to your overall test turnaround time, not just the assay turnaround time from sample to results. Because if the lab is a low throughput lab and you build your product to be running 96 samples in a row, it actually means that the lab has to wait until they accumulate enough samples before they can start the run. And that results in your actually overall turnaround time to be much longer than what you have designed for. And of course, if you build your lab, your test to be very near patient site, where you actually don't need a lab, then it could be just a single user test. So that's why throughput is part of the system that we need to consider when we talk about diagnostics tests. And finally, I think it's about data analysis and the types of testing regime that you are talking about for the assay. A lot of the tests today actually come with also a very specific software, and that would help the user better analyze data and interpret the results. We always tell our partners, the principle of your test, the technology of the test can be complicated, but for better usability and to encourage market adoption, you always want to make sure that the test readout is as simple as possible. So another consideration that I think is important to discuss is the testing regime. For example, whether you are doing single tests or pool testing, and this is very relevant for COVID management, just so that we are able to increase our overall testing throughput and testing capacity. So if you have a test that has been verified to have a certain performance only by doing one patient sample at a time, now if you want to run it on a pool testing format, you are actually looking at your assay being able to test 5 different patient samples in one well, one test. And that could actually have a huge impact to the technical performance of your test. This is because there might be an increased amount of inhibitory substances that might interfere with your assay. There might be some dilution effect on the assay limit of detection and so on. So I think these are the different things to consider. And I think every single element of this whole anatomy is important, and it has an impact on the overall solution performance. And you actually, every time when you change every single element on this whole anatomy of the solution, you actually need to go back and test verify the analytical performance of the test systematically, minimally, before you can implement it, before you can think about doing the next step of clinical validation and so on. So now I think we understand what it means when we talk about a diagnostic test. We can now look at what it means by productization of diagnostics. So the first box in this productization journey is assay optimization. Although it says assay optimization, when we look at optimization, we have to really think about optimizing the whole system, not just the assay itself. Now back to the PCR assay example. If your PCR runs on a certain instrument A, for example, then the optimization in the first box actually is to optimize your assay, particularly on instrument A. I think technical performance is a fundamental element that you will need to establish right at the beginning in order to make sure that everything that you change downstream, you always come back to test, to double check and make sure that it will not change the overall technical performance of the system. So other than technical performance optimization, when we talk about assay optimization, we are actually also referring to the optimization of the manufacturing process downstream at a very, very early stage, right at the beginning actually. So for example, if your reagent comes in 20 different tubes, and if you are able to simplify that 20 tubes into 10 tubes, while it still maintain its technical performance, then from a manufacturability perspective, of course, 10 tubes is better because that means fewer components to be manufactured, to be produced, and potentially lower manufacturing costs, and then an overall more efficient process for production at a scale-out level. So manufacturability is something that you do from the beginning of the productization as part of optimization. And then, of course, as you move along productization journey, it takes you to the really deep end of what it means by manufacturing workflow. So we look at the whole, at this process, we look at the whole entire bill of materials. We look at the whole entire manufacturing work instructions, quality control, protocols, the criteria to look at your quality control and so on. So these are the things that we do within DXD Hub for the purpose of helping our commercial partners accelerate their productization journey. Because when we work with our commercial partner, it's not just for us to license the product, but it is also for us to help them establish the production process right at the beginning of the design, before design control. So I think Anthony talked about pilot production as well. Pilot production is a very important process because at the end of the day, you do need to make sure that the manufacturing process will give you consistent technical performance. And we do that within DXD to run through a few batches of pilot production. We actually need to make sure that every single batch that we produce has the same performance, because actually one of the regulatory requirements for registration of a device is whether you are able to manufacture consistent batches that give you the same performance every time. And normally what we do is after the assay optimization, we will start with a very small scale pilot production in-house, and then with the kits that are made from this process, we will have a very small, almost preliminary run of the analytical verification. And that actually help us build and verify the pilot production, the process, the workflow, the QC that we talk about, and so on. And only after we are able to consistently produce different batches that give the same performance after the pilot production stage, then we are able to do the full-blown, analytical verification and then clinical validation. And this is when we produce at least 3 different batches. And then with these 3 different batches, we will then perform our analytical clinical evaluation using the kits that are made which will then form the actual data that will need to go into your regulatory dossier for submission. So I think this is really a deep dive that go into the nitty-gritty of what it takes to do productization, to do manufacturing. And I think it's important to elaborate and deliberate a little bit here, because one of the biggest misconceptions when we talk about productization is that people always forget about this part about manufacturing. They could have done, you know, everything else. They could have optimized the assay technical performance very well. They could have done, you know, all the analytical protocols. They could have even taken the assay down to testing on clinical samples, almost everything. But they miss out on the manufacturing. And when they come to DXD Hub and say, well, you know, we've done everything else, but how do we actually manufacture them in a mass production setting? So that actually would always almost take us back to stage one, to start all over again, to redo the protocol, to redo the assay optimization, so that we are able to build the manufacturing workflow into that and regenerate the whole sets of ABNB dossier for submission. And I think we do actually want to emphasize this point a little bit, so that maybe for those of us in the audience who are looking at, you know, doing product development for the first time, is something that is very important to keep in mind. And of course, downstream of the productization and market adoption is really a lot to do with technology transfer, tech transferring the whole manufacturing know-how, the whole product AV&V know-how to our commercial partners, and a lot to do with working directly with the end users, the lab operators, the clinicians that we talked about. So we actually go to the lab, we go to the hospital, we talk to the clinicians, we talk to the lab managers, the operators of the lab, just to get some sense of how the product is actually being used. And sometimes the end user will come to us and say that, hey, you know, our product has really been verified and validated on machine A, but really they don't have machine A on site. Instead, they have machine B, but that is supposed to have the same function. Is then very difficult for the manufacturer or the product owner to tell the lab, hey, why don't you just go and buy machine A? It's just not possible. So remember that agile iterative process that I mentioned. So then when that happened, we actually need to come back, and we would then need to verify and validate the same assay now on the machine B that is available in the lab, for example. So I think this slide gives a very pretty comprehensive overview of what are the regulatory requirements for in terms of dossier preparation, in terms of the AV&V work that you have to do, particularly for a COVID-19 assay that is meant to be used, you know, under the Singapore HSA provisional authorization, or even under the US FDA emergency use authorization, or UA in short. And I think maybe want to focus on the 3 yellow boxes at the bottom. Things like, for example, sample storage, storage media compatibility. This is actually not very obvious, but extremely important. You know, we often hear feedback about some assays that are not working that well in the lab. And in some of the cases, it's really not about the assay itself, but the compatibility of the sample storage media with the assay. For example, for COVID, there are you know, sample storage media that actually inactivate viruses upon sample collection. And some people, actually a lot of people like that idea because it reduces biosafety concern. However, some of these media can be, you know, incompatible with your assay, or it can also reduce the sensitivity of the whole system overall. So it's actually very important to verify whichever storage media that you want to introduce to the assay, to verify analytically at least that the assay is compatible with the media that you are suggesting to the end user. So another important consideration, I think, is whether you actually have tested the analytical performance of the assay in the system with respect to the testing specimen. So whether this is blood or saliva, or even saliva, there are many different types of saliva, because saliva can be Julian saliva. Saliva can also be deep throat-like kind of saliva. And you actually need to understand whether your assay has actually been proven to have the same technical performance for the various specimen that your lab will be using. And then, of course, is the platform. So different, back to the PCR example again, different PCR platform has different settings, the wavelength, the baseline, and so on. It is very different. So it's important that you must be able to, test each of the systems that you would like to claim for your product, and then to know exactly which system to use for each of the platform, so that you can then better advise your users specifically for system A, this is the setting, for system B, this is the setting, and how you would analyze the result from each of the system differently. And then again, if you think about product development from market perspective, you always have to remember that you can't really dictate what your end users will use in terms of the types of instruments in the lab. So you actually need to make sure that you give the end users as wide a choice as possible in terms of whatever that they already have. Your assay is able to work on their systems and so on. So applying this whole productization process that DXT has established over the years, together with our partners, clinical partners from Diagnostics, we were able to quickly develop and launch the Fortitude series of conventional RT-PCR assay for COVID-19 testing in the first week of February, when it was all of this happened within, I think, less than 3 weeks from the time when the sequence was available in the public domain. So today, actually, the Fortitude series of products have been distributed in more than 40 countries all over the world. And right after Fortitude, another product that we developed is the Resolute series of RT-PCR assays. So this slide shows the key difference between Resolute, particularly Resolute 2.0, and the conventional PCR test like our Fortitude. Resolute 2.0 is a direct PCR, and it detects the viral RNA that is present in patient samples. What that means is Resolute can skip the entire process of sample prep, so there is absolutely no RNA extraction. You essentially take the specimen, in most cases the UTM where they store the swabs. You take the UTM, you put 5 microliters of that directly from the collection point, and then you mix it, you add it to the well containing the master mix, and then you start the PCR process. So if you start from the first box of the workflow at the top, we are looking at the sample specimens that are either nasal, pharyngeal, or throat, or more recently saliva. Then of course, a certain transport media. For Resolute 2.0, we had tested quite a number of, actually, I think almost all the common brands of sample collection media available in Singapore and in the region. And all of these details are actually provided in the product IFU, so that the users are aware which of the ones that have been validated on this assay, so that if they are using, if they are not using any of those that have been listed, they will have to, they will be reminded that they need to do the verification in-house. So essentially, if you compare Resolute and the conventional RT-PCR, by removing the RNA extraction step, what that means is it increases our resilience on some of the supply chain, particularly on RNA extraction. Because at some point in time, I think in March, the whole world was facing a shortage of most of the well-established sample prep, the Kaizen of the world, the Biomiru of the world, and so on. And not only that, because of the shortened workflow, it actually increased the overall assay turnaround time. And that is not trivial, because as a result of that, you actually take away a lot of operator manipulated steps. You increase your overall testing throughput, test capacity, and so on. So I think maybe this is one of my last few slides. I think the development of this Resolute series of diagnostic kits give a good example of the process of agile iterative improvement for diagnostic development in particular. So this slide shows the progress of how we continue to improve as a result of the end user feedback and the evolving market needs. As I mentioned in March, when the whole world was facing a shortage in RNA extraction reagent, we together with our partner from the DSO lab, developed and launched the first direct PCR, the Resolute 1.0 assay. And then this happens in April. It's just 2 months after we launched the Fortitude kits. As a direct PCR, Resolute 1.0 totally eliminated our resilience on RNA prep for the PCR gold standard testing. And if you look at Resolute 1.0, it actually has 14 tubes in terms of reagent. And of course, very, very quickly, 2 months later in June, we came back with an improved version, the Resolute 2.0, where it now has only 4 tubes. We also then made some changes in the way the assay was done. We added differences in how the system was done. And then more recently, in July, we added another component to the whole Resolute series of diagnostic solutions, where we introduced automation. Automation is very important to scale up lab testing. Otherwise, a lot of, adding 5 microliter using a pipette seems something very trivial for scientists like us. But when you have to work 24/7, it's actually not that trivial. So remember the chart where we talk about when the sample arrived in the lab, somebody actually have to aliquot that sample into the PCR well. This is very manual. So for the sample loading step, when you are dealing with potentially infectious reagent, somebody actually need to put on the full PPE, stand in front of a biosafety cabinet, and then load this well by well. And so this is not something that is very scalable when we are looking at testing 20,000 samples a day for a particular lab. So there will be a lot of human errors due to fatigue. That there will be a lot of logistic issues trying to check what sample is added to each well and so on. So with the automation that we call RAVE, we completely take away the manual loading step. And then with this, the lab is able to run almost 24/7 nonstop. What you need is an operator who know how to press each of the buttons, and then you load the whole tube in its original UTN to the RAVE, and then the machine does all the sample loading. At the end of the day, the operator come back, pick up the plate, load it to the PCR system. So I think I would like to use this as an example to illustrate the accelerated productization process that have been developed within the DXD Hub in the past few years. With this process, we are able to actually very quickly go through not just the productization of the first product, but also to reiterate, to continue to make improvement, to push out pipeline products in response to evolving market conditions, evolving supply chain, and so on. So I think this is really my last slide. And I think after introducing Resolute series, I think I would like to introduce the teams that were responsible for developing this series of diagnostics. And this was developed by essentially 3 teams. The teams from the DSO laboratory and the DXD hub had co-developed Resolute 1.0 and 2.0. And we later on had the help of the engineering team from A-Star ARTC, who worked with DXD to develop and launch the Ruifen, our automation system to enable automation of the Resolute workflow. And I think maybe I would like to take the opportunity to highlight the very, very fundamental element, which I actually had not thought about throughout the whole presentation. And that was the fact that I think for any of this to be possible, the success of a diagnostics test really is depending on how well you work with the partners, because of the iterative process that is required for you to continue to try to improve the product so that you are able to keep to having a very successful market deployment and market adoption. And so I hope this short presentation has given you maybe a little bit of understanding of what it takes to build a diagnostic test, and happy to take any questions that the audience might have. Thank you.
Right. Hi. So thank you, Ruifen, for your wonderful sharing and bringing us through the process of productization. And also, it's heartening to learn how DXD has taken up the challenge during COVID-19 and to so that we can develop Singapore's own PCR reagent test kit. We should now move on to the Q&A session for both speakers. We do not have questions from the audience yet, but maybe let me try to start off this Q&A. Maybe this is a question for both speakers, right? So we have seen, during COVID, like a lot of emergence of new technologies that might not have been possible during non-COVID times, such as you suddenly see things like CRISPR-based diagnostics or isothermal diagnostics. So in that aspect, do you think that COVID-19 actually has changed the whole diagnostics field in some way, even after post-COVID? Just general thoughts on that.
I think in a way, yes. As you can see, at the beginning of the pandemic, when the virus was mainly in Asia, you have a supply from all the big guys, Kaizen, Roche, Cepheid. There was no such thing as reagent shortage and so on. Everybody played with the big guys. But as soon as the pandemic hit the West, immediately most of this reagent become very limited in this part of the world. Mm-hmm. And that's a time when actually a lot of the local players had an opportunity to enter the market, to enter the lab. Because for diagnostic, I think a lot of the time is about the footprint, whether or not you have a footprint. And I think it offers actually a fantastic opportunity for a lot of our SMEs who take on technology from public sectors, take on from private sectors to actually have an opportunity to establish themselves, set out a footprint in a lot of this lab. And moving on, you think about expanding the manual for the footprint that they managed to set up during COVID. And I think that likely will change how the medical lab will operate moving on, in my opinion.
Okay. So we do have a question from a couple of questions from the audience. So there's one question, this is addressed to Dr. Weng. So is there other products similar to the Resolute in the market?
I think when we launched Resolute 1.0 in April, there were no truly direct PCR available in the market. I think there was one or two in the US that had a simplified sample prep. But if you think about automation, then it is a hurdle. If you have to even do some very simple sample prep. So at the Resolute 1.0 time, I think we were the truly direct sample to PCR reagent product. By now, I think there are maybe a couple of similar products that have been introduced all over the world.
Okay. And the follow-up question on that is, are there plans to sell Resolute commercially? I mean, if you're at liberty to answer that.
Sorry, something that I have not mentioned and actually acknowledged is our commercial partner for Resolute. So we developed Resolute out of A*STAR, out of and DSO, but the product subsequently has been licensed to one of the companies, SME, Advanced MedTech. Advanced MedTech has now been the product owner for the whole Resolute series of products, together with Ruifen, the automation system.
Okay. I have one more question for Dr. Weng. So maybe this question, then before I have a question for Dr. Anthony. So Ruifen, so the question is, how were you able to convince a financial group to invest in the use of more automation and increase in support of costs, associated with production volume? Maybe this is a question that Anthony can also chime in if he's familiar with this.
So actually, something that I had not thought about is the DXD model. DXD's model is one where we actually co-develop products together with our partners. In this case, for the Resolutes, because we actually developed the product together with our public sector partners, the companies came in later when the product was already registered. Now, we activated our co-development model with respect to Ruifen. So we have to actually convince our commercial partners the value of developing the automation. Yes, it's very costly, but the value add to the Resolute series of product is extremely high. So that's the point about risk sharing with your partners. You are in this together. You put in there are certain risks that you have to take, but you assess the risk collectively, and you support the development collectively as one team. So it's a lot of risk sharing and risk management.
Do you let them contribute in terms of funding, or also share part of the technology?
Yes, so our partners contribute both cash and in kind to all products that we develop at DXD. So DXD is quite different from the rest of the public sectors. We don't actually give out grants. We fund direct project activities together with our partner. So there's always skin in the game. We put in a certain dollars, our partners will put in a certain dollar. At the end of the day, we co-own the IP that we co-generate from the collaboration.
Anthony, you have something to add?
Yeah. So for on this question, actually, from our experience is that when you are in the process of setting up your pilot production, in order to minimize batch-to-batch variation, you are really looking towards if not semi-automated systems already as a start. And so if later on you want to go fully automated, it will be, I think one way of moving forward is really that based on using your pilot production facility and processes, you are already starting to churn out lab developed tests, as in products for lab developed tests, or selling at a small scale. And with all these, the revenues coming in and then the projection, it will be easier to convince investors to pump in more money to fully automate the entire process. But having said that, there's also some other things to consider, because depending on how you automate the entire process, you may be changing the manufacturing processes and the quality of the components or the product that's come out of it. And for that, you have to do your internal testing, and you also, as the product owner, you also have to notify all the relevant regulatory authorities to make sure that they are made known of this change.
Mm-hmm. So for that process, right, when you try to redesign the whole process to become automated, that you need to notify the regulatory bodies you mentioned. So is that a very difficult task to switch over to that? You have to resubmit everything.
It really depends on which part you change. If it's like, say, for example, let me see what is a good, like maybe if let's say you just change like a dispensing machine from a small one to a larger one that has a like higher turnaround time, higher yield, then probably you just have to make sure that the sort of like the amount that's being dispensed is the same, and then there's no much changes. Then I think there's only minimum update. But if you were to overhaul the whole thing, then you really have to like submit a lot of paperwork.
Oh, okay, and that will set you back quite some time. Right?
You can start selling already, then you start to notify the regulatory authorities again.
Oh, okay.
Yeah. Then one other thing that I didn't really mention just now, I'm not sure whether I missed it, like if like Dr. Weng have already mentioned, is that one of the sort of like misconception is that we can just churn out lab like prototypes in the lab, and then we will just start to run clinical studies, and we hope that all this clinical data that we've collected can be submitted for regulatory submission. The truth is that the regulatory authorities, they really need the data, the clinical data, to come from kits or products that's being manufactured in the same condition as you are going to manufacture them later on after approval. And this is something that a lot of like the younger startups, they don't know.
It's such as mentioned about like GMP kind of conditions.
That's right. So it's important for developers, for product owners to really set up the pilot production processes, start churning out like minimally 3 batches of products already before they can start to run their critical validation.
Okay.
So maybe if I could add something on that. So a model that we have been using at DXT is when we are thinking about pilot production. At that stage, we will actually invite our commercial partner who will have a scale-up plant, or in some cases, it could be a contract manufacturer who will come in and then discuss our pilot production plan. They will come in and observe how we do the pilot production. So at that stage, we will already be thinking about how, what are the changes that we will envision for during the tech transfer to scale up, so that when you have to make changes later on at the plant, you actually won't have to revamp the whole thing. You already know what are the changes you will make. You build that into your pilot production plan. So I feel that is something that is useful.
Yes, I totally agree on that, because like if you were to build your own like pilot production processes without like getting your potential partner to come in, right, then maybe you use certain equipment that the other party doesn't have, then what's going to happen? You have to redo the whole thing again. So
exactly. So I think it's always about the whole system and about the partnership. Everybody play a part for a diagnostic product to be very useful.
Agreed.
So in that regard, is DXT like offering that pilot production or service or tie up with local startups or any even large companies here? Is this what you're offering? So
we work with commercial companies. I think about 50% of our partners are actually startups, local SMEs, and as part of our product development, like I think briefly mentioned over the presentation, part of the focus is on the pilot production and the plan towards tech transfer to the eventual plant.
Okay. So, all right, we have slightly overrun, but maybe just one last question for Anthony. So this is regarding prognostic testing. So we know that in prognostic testing, most of it is employed in the oncology fields. So in terms of infectious diseases, how do you see, there is a, do you think that there is a, because it's relatively new, do you see clinical acceptance in this area? For instance, for your dengue prognostic tests.
So for for dengue, we've been like talking to various stakeholders. For Singapore, I think with our government doing a very good job in building more hospitals, the bed crunch issue is becoming less of a problem. But for the developing countries around us, whenever I speak to them, they immediately get what we are trying to do, because they are really seeing it happening in their countries, whereby all the hospital beds are taken up by dengue patients, and later on people coming later are rejected, and then they may die outside hospital without any medical care. And I would say that with COVID-19, it will definitely become more and more of a practice, in the sense that for COVID-19, there's also a huge majority of people who are have very mild symptoms, and those who will really go into develop severe complications and really can't breathe, need a ventilator. And in areas like the US, where the disease is very widespread, how do you really make sure that the patients who really need the healthcare facility can get them, and the whole healthcare system is not being overwhelmed by all the patients coming in at one go? Yeah, so I definitely see with infectious disease is definitely something that's coming up. Yeah.
Okay, so I guess you guys are also getting approvals in the developing countries also for that purpose. Okay. Yeah, so I think with that, we have to end the Q&A session here to adjourn for lunch. For all attendees, please have your lunch break until 2 p.m. I hope this morning session has been beneficial for all. Okay. And